Hi. The question I had, I don't know if anybody else would think to ask this, but from my understanding, what I just found out is that some kids have the same, I hope I say this right, if I don't, please excuse me, variant. So the same mutation, because, this is all new to me again. I just believe I found out that one other kid, I'm not going to say who, has the same variant as my daughter. What exactly does that mean? And the variant that my daughter has, does that make it different from any other kids, actual coding, if that makes any sense? Did that make sense?
Okay. Can you hear me?
Yeah.
So I have to say, just in the last half hour, I've been rethinking this. So we all have a lot to learn is the bottom line. So as I think both of us were showing on our slides, for most individuals in the community, their change is unique to their child, right? There are two different mutations or variants that we've seen that are recurrent so far, at least in our series. I'm sure if we could get everyone in Champ1 to put their data together, they would start piling up with more than that, and you may know more about other ones as well. But certainly many are unique. I think and I apologize for getting very jargony, but I explained that there are two individuals, I think it's two, that have the same missense variant or the same single amino acid change. To me, those are very interesting from a mechanistic point of view because they're unique in that way. The other folks have truncated or smaller proteins, which might be acting in a very similar mechanism. We're guessing, but they might be. Might be some differences, but might be different. And then for three of you, you have the same truncating variant. It's truncating in exactly the same place. And so we might think that those were going to be more similar. Those three individuals that I put up on the slide might be more similar. And if we have other people out there, those might be more similar. However, and I'm not going to out anyone or say anything specific, but what I said, and I think what you were alluding to, is some of those truncating variants might actually be doing something because you showed that the protein is still there. And so they might actually, it's not as if they go away and are non-functional. They might be doing something. They might even be doing something deleterious. It might be better not to have them around. And so I think I'm still trying to figure out in my head, are there differences, therefore, between the truncating variants that are kind of small, medium, or large in terms of either their effect or what they're doing? I don't know if you disagree. I mean, this is a conversation rather than us knowing what we're talking about.
Thank you. Yeah, I think there's I remember some paper talking about the recurrent mutation. There might be some mechanism, DNA feature in that region, that can cause the same exactly mutation during the, I forgot, I cannot remember exactly what the process, but they, in the genetic field, we are starting to see more and more recurrent mutations not only in these genes and also in other genes. And in terms of the mutant protein that presented in the patient cells, we don't know. That's just our working hypothesis. They either do bad or do good or do nothing. That's something we have to figure out.
You won't know. Okay. Thank you.
First of all, thank you again for your support, everyone in this room as well. Just got a quick question for our daughter's 11, at the higher end age scale. That's and early on, I'm sure many families heard here today. Early on, when we were searching for a diagnosis, most kids go undiagnosed. Is there any way, when we talk about our sample size, even though it's large to us, of being less than 50, to get the word out to pediatricians to see if people that are undiagnosed, if there's a way to see if any of these behaviors match up? And how do you go about doing that?
So I know I said to some folks at the break this morning, but just so everyone hears, We have set up a research study for individuals with an autism diagnosis. And by that, we say anyone who's been officially diagnosed with autism to get access to the genetic testing that I think most of the families here had, that is exome sequencing. So if you happen to know of anyone in your community, the program is called SPARK, S-P-A-R-K, for autism. And with that, it's a research study, but one of the ways that we work with participants is that if they decide to do the genetic part of it, they don't have to, but if they do, and if we find a genetic diagnosis, which could include Champ1, that is on our list of things to do, those families get that back. It's all free of charge. It's only to American families. I apologize for some of our colleagues around the world, but right now it's only in America. But you can do that all from home. You don't have to go to any special place to get your blood drawn or anything. You can be able to have access to that. So for the autism part of the community, we have a solution already available to get access to that. The one other thing that I promised, and Jeff, it's going to be in your inbox when you get to it, is that many of us as geneticists in the community have written a position paper that we believe that for individuals with neurodevelopmental disorders, the first line genetic test should be an exome, and we strongly endorsed having access to that. So there's a very, sort of a esteemed, I hope, group of us who have put that out there officially. It's an official position paper from us now at this point, which applies internationally. But the point that we did it was to help get insurance coverage and increase awareness among pediatricians, neurologists, developmental pediatricians, all those folks. And Jeff is more than welcome to pass it around to everyone. You can disseminate it from anyone in your communities you know of, but it's officially out there now.
Sorry. Hi. Well, I want to thank you guys, but it goes back to the first question. So what does that mean for treatment if the kids have different variants for truncation? If we get a treatment for one of them, do we have to get it specifically for each of the variants?
So I can imagine there's a variability in terms of phenotype in all those three patients. Correct me if I'm wrong. So that's why we are trying to collect all three. For example, all three patients because they have a different genetic background, which may contribute the severity of the phenotype. Yeah. So either the more we understand that, the more we can talk about that. But now that's Thanks.
Is treatment different for the different variants, or is it the same?
So let me say a couple things, and I apologize again. I'll try and make this accessible as possible. So there are two possible mechanisms that we've been talking about. We haven't used these terms, but I'm going to use the terms loss of function versus a gain of function, right? So the loss of function is if the gene isn't working right and as if it's not there and showing up to do its job, and it's not there, it's not present. That's one possibility. If that is correct, then that means the treatment is going to be the same for everyone because we need to be able to put that gene back, a normal copy of the gene back. Hypothesis number 2 is that it's a gain of function, that the different mutations are actually doing something new or doing something bad. And it's possible in that particular case that the treatment could be different for different mutations. Possible. It's also possible there could be a generic one-size-fits-all, but I think for the research community, if you were asking me, I'd say a critical early experiment that needs to be done is understanding the mechanism of these mutations because you go in radically different directions in terms of thinking about treatment between the two of them. The other part of this, which it makes it even more complicated, is Champ1 is a big portion of the answer, but there are about 20,000 other genes besides Champ1, as well as other things that are going on, if you will, in the environment, or other things that change it. So not everyone looks like a carbon copy of everyone else with Champ1, even with the exact same genetic change, right? So there are other things, other strategies potentially to help support or correct as well.
Thank you. I just wanted to go back quickly to what you said, Dr. Chung, about the position statement from the geneticists, and I feel like if anyone in this room encountered insurance challenges with getting exome sequencing covered, I feel like that paper is going to be incredibly important. I spent a lot of time in my past job fighting for insurance coverage for genetic testing that we knew that patients needed so badly and insurance companies didn't cover it. I think at this point you've all had genetic testing, but if you have friends and relatives and colleagues who are still looking for a diagnosis, I feel like your offer to share that paper was wonderful, and it will be really helpful in sort of fighting these insurance companies and getting coverage for testing that was so important.
Hi. So I'm going back to not being one of the American families again. Skipping forward, if we do find something available for our kids, how easy is it going to be for families that aren't over here to access this sort of thing? Is it going to be sort of, I know this is a long way down the line, but I mean, is it going to be possible for families like ours? I have a fight with a daily every time I see a pediatrician, it's quite difficult to get him to do things.
So I would say, without knowing exactly what we're talking about, it's hard to answer that question. Mm-hmm. What I will say is that, so I'm going to go from a different rare disease space that some of us know about. So with certain treatments that are extremely expensive, based on the governments, they have made decisions in terms of what they can afford to treat in certain circumstances. And so I'm just putting out, this has been true for us as geneticists for a condition called spinal muscular atrophy that some of you may know about. So in certain parts of the world, the government has just decided we can't afford to do that. It's something that in US dollars cost anywhere between $1 million, well, about $750,000 a year to now $2.1 million for gene treatment. So there are some issues like that. I think some of us have made sure that we could get access for patients from around the world, and I don't want you to all come to Columbia for this reason, but we've had people from around the world come and be part of clinical trials and get access through other ways of being able to do it. Mm-hmm. So as people that support the rare disease community, we will work with you, we will advocate for you wherever you are in the world in terms of doing that, but we have to partner with you because we don't know about different social systems and social healthcare systems in all parts of the world. But I think we're committed to first getting scientifically to the point that we can do something, and then when it becomes a policy issue, we'll be with you there as well.
That's fine. I'll keep fighting with my pediatrician. Thank you.
I have a question, and it might only be able to answer theoretically at this point. But so speaking for some people that aren't in this, that didn't get a chance to make it here today, we have one family where their child is missing the Champ1 gene entirely. We have some other ones that have a microdeletion at the terminal. So those children seem to be very high functioning. And I know there's no specific answer at this point, but is there any theoretical possibilities that would make sense of that? Because you would assume, and again, I don't want to jump the gun, but the gain of function, is that a possibility? Is that what that's?
So another way of that's what we have been saying to each other scientifically, and we haven't said it explicitly. But yes, there are some individuals that are missing an entire copy of the gene, and I think it is incredibly important to both compare them clinically as well as to do some of the biochemical studies that are going to be going on, I'm sure, at CHOP and other institutions as well. To me, those are very, very high priority to do right away.
I have no comments on that. Yeah.
And I know every gene has its own, complexity in itself, but would the gain of function make it more complex?
Yeah, I definitely think so. If because patient has different form of truncated protein, and they lacking the different motif or domains in the protein, and they are made responsible for different factors come in play to be function for the complex and during the mitosis process. Yeah, I definitely think so. Yeah.
They're just, I guess the way I would say it, they're different. We have strategies for dealing with both of them, and they're just different. So I wouldn't give up, and I wouldn't say either one is easy at this point. But I do think fundamentally we have to know the difference. And it may be that there are two different mechanisms, and different people are going to need different sort of strategies to do this.
And maybe just to clarify on the idea of a complex, so just to, in lay terms, use an analogy of Legos. So for example, you take a Lego castle, and you know that there's a tower in which you have to have a bunch of gray circles or gray blocks to make this tower, right? And so what if you replace one of those gray blocks with some other shape? That would then define it as something different, right? Maybe it's no longer a tower, but now it looks like a wall. So that's the difference here, is that when you lose those partners in which Champ1 is interacting with, maybe the function will be completely different. And I think that's what's getting at what they both have described, is that the interventions may be required, different interventions may be required. Maybe. Right.
Hi. I have a question for our international folks here. I just touched a little bit on what Stacy was asking. They have I don't know how to say it. Not a company, but a sort of like a Europe group that champions and advocates for the rare disease people. My question to you guys would be, the Simon Searchlight, have they ever worked with international groups like this? And if so, what are the barriers, and what can we do to remove those barriers so that this particular group, URDIS, can advocate for our Europeans family that want to share data and enrich our data bank?
So I'll speak only on behalf of Simon Searchlight in terms of this. So Simon Searchlight, unlike what I was saying with Spark, Simon Searchlight is open to anyone around the world in terms of doing this, so there's no geographic barrier. For those of you who may have seen Lindsay earlier, one of the things she's been letting people know about, we're translating materials into several different languages, and within the languages, French, Spanish, I'm going to get them wrong, Italian, Dutch, and I'm missing one other, are priority languages in terms of doing it. So it is heavily Eurocentric, I think, in terms of those countries. Biospecimens are a little bit logistically more difficult to be able to get from Europe to the United States quickly while the cells are still living, but otherwise in terms of data, it's all definitely depositable. In terms of any sort of collaborations, what I'd say is anything that's in Searchlight, again, is freely available to any individual researcher, any collaborative group, I don't care, anyone who can use the data responsibly, I'm glad to give it to. And so I'm glad to do it in that way. I'm glad we've had other consortia, European, US, in terms of rare diseases and putting all our heads together scientifically on that. So like I said, we're open to any, if you want to put us in touch with colleagues, if you want to just let other colleagues know the data are here, they don't have to collaborate with us, they can just take the data and do good stuff with it. But we're glad to do any of that. So there shouldn't be any barriers from our point of view. I guess let me just follow up on that, though, to say, and I'm just going to give you a couple words of advice, because all of us have been involved with many rare disease communities. First, just a shout out to Jeff and everyone who's made today happen, because I know it's been a labor of love and a lot of hard work. This is the first meeting, but I'm sure it's going to be one of many, many. I know there are many people who couldn't even physically be here, but the great thing is that you've got the internet now to be able to bring you together as a community, so you can get together as often as you like in terms of that. So I'll just put that plug in. Number two, stick together. You're a small community, and even though there are many other people out there not diagnosed, you're never going to be incredibly common, and so you're going to need your numbers to be able to stay strong and advocate for yourself. So I'll say this, and at some point it will come up in your future with someone who wants to splinter off, and I will just urge you not to splinter and to stay together united, and for each of you to stand up for yourselves. And what I mean by that is that you've got to have your family represented, your child represented in the research. You need to contribute the data. You need to tell us how to make it easier for you to do that, but you also need to do your part and contribute the data. And then we need to help you by giving you good evidence-based information so that you can then advocate for your child and so that your providers will listen to you. Right? We've got to do this together, and we're willing to do, as professionals, we're willing to do our part in this, but I want to say you also have to do your part in this. We're going to try and make it as easy as possible, so it's not more than a couple hours every once in a while to do it, but we do need you to do that because that's what's going to be best for your child in the long run. And because many of your changes are unique, you have to stand up for yourself. If you don't stand up for yourself and you don't have yourself be counted, I can't guarantee that any scientist is going to work on your family, your child's condition. And as we've been talking about, it may be that there are differences between the mutations. So if you want to make sure that you're doing best by your child, you know what to do.
I can ask it after. You go ahead. Sorry, going back to the loss of function, gain of function. So if Champ1 ends up having a gain of function and the kids who have a loss of function are higher functioning, is there a way to get that to completely shut off and create a loss of function in our kids that may help them advance? And what would that look like, I guess?
So I'll give my opinion, you can give yours. So very briefly, there are ways of being able to use what we call oligonucleotide therapy. That's one approach right now. There are other people that would do it with using a vector and putting something in to deliver into the cells. But basically, there's a way that you can gobble up the bad gene, degrade it, get rid of it, and that would then change it to a loss of function. But to convert it from a gain to a loss of function, that might still not be ideal. Might be better to do a replacement, just an out-and-out swap out, but that may be a longer-term solution. I don't know.
Yeah, that's all. Yeah, yeah, small RNA to target the mutant allele, to get rid of it, and that'll be.
That seems like an easier solution, immediate solution.
Yeah, I think a couple, company working on the different disorders, but to use the same, strategy for that. Yeah.
So just to caution, when you say immediate solution, this is not going to happen tomorrow, right? But right. And I think echoing what Wendy was saying is that basically you could have that bad RNA gobbled up or whatever, the bad protein gobbled up, but then is that better than basically putting back the normal protein? We don't know. Yeah, right. And that's why these models, these tools that we use to test these different hypotheses are very important. They're critical. But that also comes in part through your participation as a community to inform us of what we need to be doing as researchers, right?
I want to be sure I understood the question. How do we advocate on that front in terms of? Right. So just in case people aren't on camera, I'll repeat the question. So how do you advocate for yourself and make sure that your voice is heard is to make sure that however it is that you guys are collecting data, it could be Simon Searchlight, you could have other ideas, but however you're doing it, make sure you're putting in your data, make sure in terms of the cell lines that are being made, make sure cell lines are being made from your child, make sure you've donated that skin biopsy blood sample, whatever it is that needs to be done for this. Hopefully at some point there are going to be IPS cells on every one sample that's put in, make sure that yours is amongst those that are made into the IPS cells. Interestingly, at some point, and we're going to hear about it in just a second, the mouse models are an issue. It's going to be hard to make a mouse model for every single mutation. It's just prohibitively expensive. And so at the beginning, though, I would guess there are going to be a few mice, and we'll see after we see what the results of those, how many more we need to make.
And I guess what I hear you guys saying is that we'll know more information in like 3 to 5 years. Is that what you're saying in terms of whether we need to keep the other Champ1 gene there, or if it'd be better to remove it? I guess the timeline is approximately 3 to 5 years. Is that correct?
Thank you. Yeah, yes, I think so. Dr. Harkness just said that, and as you know, the research will take time, and especially the IPR cells will take more times than other experiment because it's, sometimes you get the contamination and you have to start again from the very beginning, and you have to select the cells, make sure they have all those markers, cell identity to make sure they are the cells that you are looking for. Yes, a lot of back and forth steps to make it happen.
Up the process, is that what you're saying? And then also financially, are there things we can be doing to help speed up the process?
So I'd say absolutely. As a community, I think what you can do is for the people in the room, the people who are online or that watch this later, the more that you can do in terms of just making sure that everyone is part of the research and part of that solution, that's the most important thing that you can do, and it won't cost you a penny to be able to do that. A little time, but it won't cost you anything. One of the things that I don't know if you appreciate, I can't always tell what's going to be my aha moment. I can tell you I've had a couple already this morning, but I don't know until I talk to you guys. And so I just can't even always know what questions to ask you. It's just listening to you and watching your kids and being able to see the data that are coming in. But it's just so important, therefore, for you to be able to interact with us, and that's why we have these meetings like this, as well as to be able to put your data in. So that's the first thing. The other thing is in terms of advocating. So again, this doesn't cost you any dollars, but I urge you guys to think strategically about how you do things that don't cost you anything, but make sure that your information gets to as many researchers as possible in a way that they can digest it and use it. And so I stand up here today mainly to be able to say I will always do this in a way that it's not my research in terms of doing this. It's working on your behalf to make sure more people get access to your data in a responsible way. I will always commit to doing that for you guys, but you need to do things that are going to do it in that way because you don't want to do this thing over and over again. It will never cost you a penny. I will always make sure in terms of doing this that your foundation, your group is never charged for this. It's always accessible to anyone doing it. So that, again, that doesn't cost you anything. If you want to be able to advocate for yourselves again from talking to your congressmen, your senators, anything else that you can, again, the National Institutes of Health are the main funding agency that funds biomedical research in this country. For those of you in other countries, you know your funding resources for that. We are constantly under attack, and I have to say one of the things I'm always worried about right now in Washington is that the administration, the presidential administration, wanted to cut the NIH budget by as much as 40%. Which would have been catastrophic for biomedical research. So anything you can do in terms of continuing to let your congressmen, your senators, as you vote for doing this, I'm not saying who you vote for, I don't care, but just in terms of how important biomedical research, 20th century cures, those types of things are. Again, it doesn't cost you anything, but you can do those. And then finally, as I said, funding can come from what you guys are doing in terms of fundraising and being able to give research grants and things like that. I just urge you to think about what are the key experiments that you need to do that are going to be catalytic for the field and that are going to bring more researchers into the field. Because you're not going to have a lot of money, unless one of you is a private bazillionaire that I didn't realize. You're not going to have a lot of money, so you need to think about what's really critical in terms of what's important for your community, as well as scientifically, what are the big inflection points that are going to change the research direction for the group. So, and we're all here to help you, right? So if you need advice, if you need help, we're here to give advice in any direction you like.
One question I had, all this genetic manipulation stuff sounds incredible, but are there any inherent risks that, or that are increased risks for our kids? Like, let's say you guys find something and we change the DNA, and then is there an increased risk of cancer or, other things that we kind of need to consider as we're thinking about these options?
So I'll give you one consideration, and I don't want people to panic. So let me underscore this. I don't want people to panic. As I have been thinking about Champ1 and knowing what Champ1 does, one of the questions that's been in the back of my mind is whether there was going to be an increased cancer risk for this community, because there is, I can make up a scientific story about how there could be an increased cancer risk, but you don't know until you see the data. As I said, our community is young, and cancer for the most part happens in older people, so it's a type of thing that I wouldn't expect to start seeing increased cancer risk until, in general, people were in their 30s or 40s or 50s. One of the things that I do want you to, again, I don't want you to panic, I don't want you to run out and get MRIs head to toes of your kids, but it is one of the key data points that I'm waiting to see greater numbers and longer periods of time in older people to answer that question. So that's separate from the therapy question you were asking, but that is to me a key medical question that we need to think about. The second is that, yes, in terms of gene therapy, we have a long history of gene therapy internationally, and we do have cases in which, I won't go into exactly why, but we have induced cancers in some people. We have caused death in some people with these treatments. And so whenever we're thinking about anything like this, we are extremely, I think you mentioned this earlier, we are extremely careful about this. We do lots of studies in mice, in non-human primates, in cell lines, in all sorts of things, because we never want to do any harm as we're thinking about this for any individual. With this, what I will say is that the newest gene replacement strategies are much safer, but still, I would say we don't have long-term outcome to know about both safety and durability, whether it continues to work in the long term. So I think these are still unanswered questions. We keep working nonetheless, though, and I think we need to just be honest with each other about what we know and what we don't know and try and fill in as many gaps in our information as we can.
Hi, I've got another question. So with regards to the age of the kids, we do have a couple of older champs in the group. I think we've got like she's in her 40s now in the Netherlands. So I've got a 12-year-old, as I said earlier on. With regards to doing the gene therapy or any sort of treatments and things, is there like a better age to do these sort of things on our children? Do we catch it younger and there's more effect with it from being from a younger child? Or, because I mean, Shelley and I were just talking, I mean, we've been doing this for some time now, do you know? How have you seen anything within other communities, that therapies that have worked? How did that change the child? I can't imagine Callum being anything different to what he is right now. I would like him to be a lot calmer. I would like to not have to pull him by the ankles along the hallway when he lies down and I can't get him up. So I just wondered, what sort of changes have you seen within older people within different communities, if you have?
So personally, I would say we are in our infancy, almost embryonic, even before infancy in terms of this. As a general rule, and I think Håkan Håkanarsson was saying this, the earlier in general that you treat, I think our thinking generally is the better, right? There are important experiments that need to be done about what I would call reversibility or window of treatment. We're thinking about how to do those in mouse models. We do have ways of being able to make mouse models where you can flip genes on and off or flip mutations on and off. And to me, that's a critical experiment to know whether or not for Champ1 and the different types of mutations, what the window of molecular treatment is going to be. We haven't even begun to do those experiments. And so I think there are a lot of unanswered questions at this point. I think in terms of, I don't know that people appreciate it, but I said very specifically, I don't know that we're going to come up with a cure with a capital C in terms of this. I think about how to improve quality of life and how to support individuals, and that may come in different flavors for different people at different ages and with different circumstances. And I think that's, for me personally, what I'm committed to doing. As much as we can do, we will do.
Yeah, I mean, that, to me, that's what I would sort of envisage. I don't see Callum being very different to how he is now, but if there's something out there that can help him sort of just cope with life on a daily basis a little bit better. The other thing that I had written down as well, do you think there's like a spectrum? You know, you've got the autistic spectrum. Do you think there's like a spectrum of Champ? Because looking, obviously, it's great that we've all got our kids together today. It's hard to judge on Facebook how someone's, not judge how kid did, but how people are affected with their children. So personally, I think that there's very high functioning champs, and I think we've got other champs that are very low functioning, and they've got a lot of other extra pressures on their family because they've got it. I mean, we've all got it tough, but they've got it extra tough, I would say, with a lot of things. So is it possible? Jeff and I met with Professor Kind, Peter Kind. Well, we Skyped him, sorry, and he was saying that it could be sort of environmental. Like, I don't treat Callum any differently. I just treat him as he is. If he's been bad, he's told he's bad. Do you know? So do you think that helps? Is that, like, I'm just thinking for other families. Do you think that the environment that the child is brought up in and has a factor, or is it the actual gene that's making them be that way? If that makes sense.
So I can think of at least 3 contributing factors, and I'm sure there are more. So I think there are different changes within the Champ1 gene. I think that's number 1. I think number 2, there are 20,000 other genes that contribute. So I think that's the second. And then I think the third is that there are what you're calling environmental, but there's, to me, that's everything else that's not genetic is basically what it comes down to, right? So everything from in utero to early after birth, throughout life, life circumstances, what happens, infections, diet, upbringing, educational opportunities, siblings, friends, I mean, all sorts of things. And so I think it's just really, really complicated. So the answer to your question is yes. For other diseases, I'm not going to dishearten you, but for other diseases that have complexity, we scientifically have tried to dissect those and understand what the contribution of those are. We oftentimes require sample sizes that are in the, say, 500 plus to be able to start really understanding those, and that's just starting to understand those. We're right now as a community, I would guess about 50-ish in terms of this. The other complexity over this is that, I think you alluded to it, but right now we have what I'll call cross-sectional data. So we have individuals at different life points, and there may be different things that happen at different stages of life, and so there is that variability as well. So it's going to be enormously complicated. It is, I'll tell you, and many of you have alluded to this, it's a numbers game. Right? And in terms of the numbers, part of why many of you correctly identified we have to get more champs out there identified is because it is a numbers game. And so that's why many of us, you as well, are correctly identifying that as an important bottleneck to deal with.
Thank you.
And we could just take about 2 more questions.
This is Ashley's question, so I'm gonna ask for her. Yeah. Okay.
So my daughter will take her hands, clench them in a fist, cross her legs, and kind of like tense up. We've gone to the doctor, and they classify it as infant gratification. I'm wondering if anyone else's children do that. Is that a part of Champ1? Is that something else? Just kind of curious.
Yeah, Callum does it, yeah. And yeah, I've been told that it's nothing to really... Yeah, yeah.
Earlier today, she said it was more sensory. She believes it's more sensory.
She was doing it earlier when she was going the other way.
Just to make note that in the breakout sessions, we're going to sort of have these family group discussions about these topics, behavior and neurological symptoms. So I'm not discouraging you guys from talking, just to let you know, so because we're going to need to move on in just a second. Do you guys have questions?
So I don't know if you guys can answer this, but I know we're talking about, like, in theory, right, in the future. So what are things that we can do now that will help our kids? Like, what therapies, what things will benefit them, like speech therapy, or if there are any specific things that we know for sure will impact them in the future that will help them be of greater function or that you have seen? I know that we talked about glasses, we talked about, but like PTs or OTs or different early interventions that you have seen that could benefit our children.
So I'll tell you, this is what I would recommend for each of you. So to me, language is really important. I think as many of you appreciate, your kids understand a lot, but they're not able to communicate as effectively. There is a different portion of your brain which is involved in music than it is in language. And so one of the things I actually have with our kids is that we bring music into this in terms of learning to speak. So as much as you can, and so music with lyrics, so singing in terms of this, and that actually activates additional portions of the brain that help in terms of learning those things. So I know it's perhaps going to drive you nuts to have the same toddler tunes over and over and over again, but definitely do that. Have your children be very stimulated in terms of language. I don't care if you talk to them about baseball, astrophysics, gardening, bird watching, I don't care, but talk to them about it, show them pictures in terms of so they know what you're talking about. When they're asking for something, as much as they can, try and make them make an utterance of the word. It doesn't have to be perfect in terms of that, but make them work for it, as you're doing this. And I know it gets frustrating, but do those little things. If there are particular times where they need something besides using their words, and some of your speech therapists will vehemently disagree with me, and I totally understand that, but I do think assisted communication can be helpful, especially if your kids are getting to the point where they're tantruming and they're just having meltdowns because you cannot understand what they're saying, and it becomes a life unto its own. And so whether it becomes gestures, signs, picture cards, communication devices, other things, you can do that. What I will warn you about is for parents of young children, many times our communication devices are on a tablet or an iPad, and that now causes great confusion for the children because they associate an iPad or a tablet with fun. So it becomes games, fun, videos, things like that. And they don't want that to be a communication device, or they have difficulty distinguishing from that. So if you have really young children and you're planning, you're thinking about doing it, you have to, in my opinion, start from the very beginning that that tablet for you is going to be a sacred tablet for a communication device, and it does not become a mechanism in terms of entertainment or videos or games or things like that. So those are my sort of practical tips in terms of this. As you're doing it, you try and get as much therapy as you can. The more therapy you can get, the earlier you can get it, you can. Speech therapy, physical therapy, special education, fine motor therapy, occupational therapy, hippotherapy, aquatherapy, whatever is in your neighborhood that's good, but all of those are things that can be helpful. Socialization is important, and I know I had this discussion with some folks. I personally, if you have the opportunity to have your children in some way in an inclusion classroom by the time they get to school age, I think it's important both ways, both for children with disabilities to mimic older children without, or children without disabilities, and I think it's important for our community of quote-unquote neurotypicals to be able to understand how to deal with individuals with special needs. So that's just my own personal opinion in terms of doing this. And as parents, you should not be ashamed or embarrassed to advocate for your child strongly in terms of their individual education plans in the United States. And to the point, go in prepared as you do it. Learn everything about how the school system works, your IEP works, transitions at 3 years of age. Do your homework in terms of doing that. Go in knowing what it is that you think your child needs. If you have to get your child independently assessed, aside from your school psychologist, because you don't think your school psychologist is doing it appropriately, do not be afraid to do that. And go in prepared with the data that you need and come in with the plan that you think you need. Hopefully everything works well and you've got a good school system that will be able to accommodate your child. But if it's necessary, and very rarely I find it is, but if it's necessary, you may have to go even beyond that. So you may have to advocate from a legal point of view. But it's your child to do what you need to do.
Yeah. Can I ask one too, since I have the microphone? I have the mic. No. I do have a question. When you guys do your research, do you look for treatments through like natural supplements too, or is it mostly like like pharmaceutical drug?
I mean, the more we understand the mutation does, we have a better understanding what we are looking for, and we are not restricted to the FDA approved drug, but that's a starting point, I think.
Just to add to that, so I met a foundation head who they said L-carnitine. I tried it with my son. It didn't do anything life-changing for him, but for their group, especially for the younger kids, it was life-changing. And I believe Dr. Harkin-Arson mentioned something about an amino acid that was helpful for a group.
Oh, that's a TBCK case. That's a gene involved in the mTOR pathway and the leucine. You can order actually from Amazon very easily, and then that helps. At least in our in vitro cells model, we can see that rescue the mTOR activity.
And I'll make sure everyone gets questions. I just want to add to this too. Gad, sorry.
Oh, just to add to that. So I think to clarify that specific point, there are some conditions that are basically based on lacking a certain type of enzyme, for example, or some kind of, I would say, component that could be provided by a supplement. And then you get correction of some of those problems, right? But we don't know if Champ1 is actually related to that. So the research that people do would be of any, I would say, flavor. That might include that, but also drugs, certain types of compounds, these ASOs that we talked about earlier, and gene therapy. So,
currently, I think there's no evidence of a corresponding enzyme from what we know. Talking with a group at lunch and just my own personal preference, I would do anything that was safe and effective for my kid. But personally, that would be like a hope or a dream that there would be something natural that would help. So my question is, just talking to so many different people and families just like me, high throughput screening, which is an option, small molecule to upregulate the good working gene. I know it's being done with libraries of tons of pharmaceutical or already FDA products. Is there really a market, or has it been tried with large plant-based products, just all types of natural stuff along with the FDA approved products?
So I'll say there are groups. NIH, in fact, has a group that does high-throughput drug screens, and it's not just around FDA repurposing of drugs. So it does include things that are essentially pharmaceutical companies could care less about because they're natural things. They're, so yes, there are groups that do those types of screens. It's a complicated issue, but to do any high-throughput screen, you need a high-throughput assay to make sure that you can see whether or not the drug or the compound is having an effect. And I think that's one of, again, if you were to say what's a high-priority item for the research agenda for the group, that's a high-priority item to get you to the point of even being able to do a high-throughput drug screen.
and in terms of research agenda for the group, I think it would be worthwhile spending some time formulating a strategic roadmap for the foundation and what the community would like to achieve over the next year or 5 years, some defined timeline. It sounds like there's a need for that kind of conversation, and maybe there's some time set aside tomorrow for that.
Sure. We'll make it around. We'll make it around.
Hi, we're talking about just alternative treatments and ways to help our child in the meantime. What are you guys' thoughts about stem cell treatments? Like, we saved our umbilical cord, our umbilical blood, we banked it for our daughter, and we potentially wanted to use it for JJ. Do you think stem cell treatments would help when it comes to Champ1?
So I'm going to give you a very strong answer on this because I think in the community right now, there's been some confusion and misguided advice. So just to be clear in terms of this stem cell treatment, the idea is that you would give stem cells to an individual. They could either be from themselves or they could be from someone else, and they would somehow repopulate the cells in that person and be able to correct the genetic issue that's missing. So if you were to take, and so we sometimes collect these stem cells as cord blood samples and bank them, and that may or may not be what you're talking about. So I won't name the group, but there's a group in the United States that's been doing this for children with autism or other other neurodevelopmental disabilities and trying to use stem cell treatment in that. The evidence to support that being effective is basically zero. It is not effective. It's not curative. I have had patients that have gone to all parts of the world to try and get this done, and I can tell you it's not been effective, and in fact, those stem cells do not engraft. They do not stay in the individual, and so it's been spending a lot of money for nothing, and it has the potential to actually do harm. So absolutely not in terms of those types of circumstances. Should you do that, waste your time, waste your energy, waste your money in terms of doing anything like that.
All right, thank you.
Hi, I kind of just want to, because I had a great conversation with Dr. Chung over lunch, and I just kind of want to echo a little bit about what you talked about and the crux of what is being said here. Everybody wants to help, but the best way we can help is by getting data. So when we're online and we're talking, it's not just Jeff or Stacy, it's everybody that's a stakeholder in this. There's garbage in, garbage out. We give them weak data, you're going to get weak results. So you want to make sure you give them all the data and we have all the people participate as much as possible. And secondly, my understanding is there's a big research aspect to this, but it's also taking that data and transforming it into workable knowledge. So we talked about when a child gets diagnosed with Champ1, there is no book right now on how to handle this. The data they retrieve from us is how they're able to tease out what the best treatment and the best course of action moving forward. So we talk about somebody that's like Caleb that might be too old for these therapies, or we don't know where we're going to be with this therapy. That still doesn't help us in the immediate future. Having data and having workable knowledge so that we can tease out what is a false signal and what is a correct way to go is how I am understanding you guys. And I just wanted to speak to our community here that's made it today so we can reach out the other half that couldn't make it for whatever reasons. I'm not saying anything negative, but we owe it to ourselves to police each other, to give them robust data so that we can get robust results back. Am I making sense? All right. Yeah.
It's okay. Hi, I'm so sorry. Apparently, last question. I heard you mention earlier with the communication device, because Avi has one, but she's very limited to certain therapies now that she's a little bit older. And I noticed, like, it's funny, in the other countries, they get tons of therapies we never even heard of. So obviously that's why we're super grateful to be here, because we learned so much in a shorter period of time. And I was wondering, how do we go about pushing for Avi to do more therapies, or therapies we never heard of, because we just obviously found out a lot today. And her insurance, is not limited, but, some people are like, oh yeah, once a week, but other people, that I sat and spoke with, like I said, it was amazing some of the stuff that they get. How do we push for that unless it's not in our area?
So I'd say two things. Number one is that ultimately what is most convincing to payers in this is that there's evidence to demonstrate efficacy. That's sort of the gold standard that we have in this. And this gets back to, again, we need to put our data together to demonstrate that it really is effective. That's a longer term thing, but ultimately we have to do that. In the short term, what you need is someone who can be your champion and work with you in terms of being able to help you with the letters of medical necessity and being able to give a good justification for why you need the prescribed treatment plan. And so I'll just point out that if that's me, that's okay. I'm happy, since your daughter's my patient. So I'll be happy to do that. But if there's someone who knows your local resources better than me, your pediatrician, neurologist in Pennsylvania, something, that's fine too. There are certain things that you may hear about today that are just not covered by anyone's insurance, and that's just a fact. And so I can't promise that any of us are going to be able to get you everything that you might hear about today and want today, but basic things like special education, speech therapy, occupational therapy, physical therapy, that shouldn't be a problem to get optimized for what she needs.
Thank you so much. And I wanted to just figure out with her communication device, it's like, I feel like it's, well, I guess that's the whole point of this. It's like kind of limited, if that makes sense. And I feel like we're constantly trying, well, we're her voice. We're the ones pushing for everything to be so she can have the best ability. What other methods, I don't know if someone asked this question earlier, what other methods can we do at home? Because I'm very hands-on with her, and I try to, honest to God, go out and buy whatever I can afford, but as much as I can do at home, what she gets in therapy, I do a lot at home. But like I was saying, what other methods can we do to better, just for right now, because theoretically speaking, you said that won't be till a couple of years to possibly getting something. But what can we do right now to just normalize, her for right now, if that makes sense? Sorry.
So unfortunately, I've given you my wisdom so far. I don't have a lot of general advice until we get to individual sort of patient visits and doing those types of things. And as I said, if you need to, let me know. And as I said, we'll get Diane into the picture and we'll put together a package. What I would say that is, I don't know, Jeff, do you have any therapists actually here at the meeting to help in terms of giving specific advice? And
Yeah, that would be great.
yeah, okay. So I think this is to come. So stay tuned. Is that right? Part of the rest
Yeah,
of the
so in our breakout sessions, there's going to be a behavior analyst, a child psychologist to kind of facilitate the behavior session, and then there's a neurologist supposed to be coming from Arnold Palmer to help with some of the neurological seizure discussions. We could take one more question if anyone has one.
I was wondering about and I may not be saying it right, but the pharmacogenetic testing. My daughter does not respond, I guess typically, to a lot of pharmaceutical medication. Would having that type of testing done help in research for her case?
I'll give you my opinion, and others may disagree. But so pharmacogenetic testing is completely different from the Champ1. It's even a different type of test in terms of when your doctor orders it. It's still a blood test. It's still about genes, but it's a very different type of thing. The way we are as a community, the pharmacogenetic testing generally, it's specific for each medication because it's telling you how that sort of medication is metabolized and broken down for the most part. And so it's not like you order just one. It could be actually a series of pharmacogenetic tests that you have to do. For the most part, for the medications, at least I know your children to be on, they're not strongly governed by pharmacogenetics, and they don't have a very tight window in which we need to get the dosage just right based on their genes. So my gut feeling is it's probably not going to be useful. And I have to be totally honest, most doctors, if I gave them your pharmacogenetic results, wouldn't know what to do with the information anyway. But
I'm sure not. Thank you.
Thank you guys so much. We appreciate your time and all your help today.